Hi everyone, this is the second lecture on Chapter 12. Chapter 12 is on host defenses. Specifically, we did an overview in the last lecture, and now we are on to the non-specific defenses. In this mini lecture, I will be talking about the second line of defense, and in this case, phagocytosis. So with the second line of defense, this is that generalized and non-specific defense that the body will do in case of infection. And there is support and interaction with the specific immune response, which we will get into in Chapter 13. The second line of defense is made up of 4 parts: phagocytosis, inflammation, fever, and antimicrobial proteins. In this particular lecture, I'm going to talk about phagocytosis. And it's the cornerstone of inflammation and specific immunity. It's the key here. So the general activities of phagocytosis are really 3-part. The cells within phagocytosis, which are the professional phagocytes, neutrophils, monocytes, and macrophages, will survey tissue compartments and look for microbes, particulate matter that shouldn't be there, and even look for injured or dead cells. Its second role would be then to ingest and eliminate these materials, and the third role is to actually extract immunogenic information from microbes. So things that would make them recognizable in the future, phagocytes can actually do that and store that information. So let's look at the different types of cells that are considered phagocytes. The first one are the neutrophils, and these are our early reactors in the inflammation response to bacteria or other foreign materials, and also to damaged tissue. These are the ones that you're going to see as the first responders. Often we look at the neutrophil count in the blood to look for a bacterial infection. A high neutrophil count would be a common sign of bacterial infection, because we would want lots of neutrophils to fight that infection. And then when you have pus on an injured area or infected area, the primary component of that pus would actually be those neutrophils, which are fighting the infection. The next 2 cell types are monocytes and macrophages, which are actually basically the same cell. Monocytes will be transformed into macrophages after they have migrated out of the bloodstream and into the tissues. So it's actually a maturation process. Monocytes become those macrophages, and then they're ready to participate in phagocytosis. So what are the mechanisms of phagocytosis? There are 3 main parts of it. The first is that it must recognize something as foreign, then it must engulf that foreign part, and then it proceeds to kill it. So phagocyte literally means eating cell, and so phagocytosis is that physical process of engulfing a foreign cell, attacking and dismantling it. And this can be an isolated event. It may just find one foreign cell and actually get rid of it, or it can be part of an orchestrated event of inflammation. So if there's a little bit bigger incident in the body, it can be part of that inflammation process. Excuse me. So what events take place in phagocytosis? What we see is that there is chemotaxis. This is the actual attraction of phagocytes to an area. They're actually chemically drawn to it because there is either an infection or some sort of foreign particle that has been detected. The ingestion of that foreign cell or particle. And then once it does that, the phagocyte has lysosomes as part of it, and it will form a phagolysosome formation. This is where the lysosome then can actually start to destroy that foreign cell. And then the last part of phagocytosis would be the excretion of the destroyed cell. So how does it recognize a foreign invader? How do phagocytes do that? And they do this through looking for pathogen-associated molecular patterns, and these are referred to as PAMPs. These are signal molecules that can be found on microbial surfaces, and they're recognized by phagocytes and other defensive cells as foreign to the body. So the molecules are shared by many different organisms, particularly in bacterial cells, but they would not be present in a mammal, so they're easily detected as foreign. They serve as that red flag for cells of the innate immunity, as something that is not self. So in bacterial PAMPs, or bacterial pathogen-associated molecular patterns, mainly we look for peptidoglycan and lipopolysaccharides, which are found in bacterial cell walls but not found within the host cells. And then for viruses, the viral PAMPs are double-stranded RNA, because naturally occurring double-stranded RNA are found in viruses but not in mammals. So these are those pathogen-associated molecular patterns, or PAMPs, that these cells are looking for. So they're looking for something foreign. And how they look for that is that they have what are called pattern recognition receptors on these phagocytes and other immune cells. And these PRRs, or pattern recognition receptors, recognize and bind those PAMPs, so they're specific for them. They'll actually recognize and bind them. So cells that possess these pattern recognition receptors will recognize these PAMPs whether or not they've encountered them before, because they're specific for that actual structure. So that's actually how these immune cells will recognize and attach to the cells. And after that, what we get is phagocytosis. And this is a great table or great figure in your book to look at. This is table 12.1, and it goes through all of those steps of phagocytosis. So we have chemotaxis here, that it's being drawn to this by some sort of chemical signal. It will come and do adhesion using its receptor cells to adhere to the bacterial cell in this case. It will do engulfment and then form that phagolysosome. When it forms that, it will actually start to destroy the foreign cell. And last, we have elimination. This is actually a really great table to look over. It talks about all the different steps and players. So I encourage that you look at this, and this will also be a good source for our wiki page this week if you are one of the people who are responsible for a step of phagocytosis. So hopefully this is a good overview of phagocytosis and the players involved, and also the steps involved in the second line of defense. In the next lecture, I will be talking about inflammation and fever.